Avian pioneers precision oncology in lung cancer and infectious disease solutions. First, integrity. Second, professionalism. Third, innovation. Lastly, sustainability. Our key pipeline focused on lung cancer treatments. MEKI for NSCLC target MET is currently in phase 2 clinical trials globally. ABN 202 for NSCLC is currently in the discovery phase. ABN 501 for SCLC targets Claudin 3 and is currently in preclinical stages. The advantage of our main pipelines. Abamekib has an exceptional safety profile, promising anti-tumor activity and combination therapy advantages. ABN 501 is an exclusive anti-Claudin 3 antibody with a high specificity, strong affinity, and promising target for SCLC. ABN 202 is an antibody cytokine fusion protein with superior efficacy and broad application, synergizing with immunotherapy. We believe it is positioned as beyond ADC. Our ABN 202 offering superior efficacy with a selective anti-cancer effects, having synergistic effect with the immune checkpoint inhibitors, and showing excellent result in ADC-resistant models while reducing systemic toxicity through inhibiting cancer cell growth via interferon beta signaling, activating immune cells and leveraging ADCC and ADCP functions. ABN 202 targets EGFR, TROP2, HER2, and bispecific antibodies, and it's aiming for IND submission in fourth quarter 2026. ABN 202, depicted on the left, uses an interferon beta mutant enhanced through glycoengineering. Avian is developing interferon beta mutant to be active in the tumor microenvironment, such as in low pH conditions. This newer version highlights systemic toxicity issues found in existing cytokine treatments. Beyond ADC, ADCs can lead to drug resistance. However, ABN 202 induced apoptosis by type 1 interferon signaling pathway, which maintains strong efficacy even in ADC resistance models. Even in cases with low antigen expression, ABN 202 shows better results than traditional ADCs. Conclude our Beyond ADC therapeutic, ABN 202 is expected to be more effective in patients resistant to ADCs and those with low antigen expression, overcoming the limitations of traditional ADCs. ABN 202 demonstrates strong anti-cancer effects by inducing apoptosis and boosting the immune system. ABN 202 can convert cold tumors into hot tumors, which respond better. This makes ABN 202 an excellent candidate for combination therapy with immune checkpoint inhibitors, resulting in enhanced clinical efficacy and multiple anti-tumor mechanisms. The ABN 202 platform is being researched and developed with monoclonal and multispecific antibodies for various tumor antigens. ABN 501 is a fully fucosylated human antibody with high specificity and strong binding affinity for Claudin 3. Claudin 3, the target of ABN 501, is typically located at the tight junctions in normal tissue, but becomes exposed during tumorigenesis, resulting in high expression in various solid cancers. Therefore, ABN 501 is both safe for normal tissues and highly effective against multiple cancers. Currently, preclinical toxicity studies are ongoing with the goal of submitting an IND to countries including Korea and the US by the second half of 2025. Small cell lung cancer is a key potential indication for ABN 501. There remains a significant unmet medical need for new drugs. Claudin 3 showed the highest average expression, indicating strong expression intensity. Claudin 3 has a significantly higher expression rate and presence in both limited stage and extensive stage SCRC. ABN 501 is effective in small cell lung cancer cell lines. Based on these findings, we have partnered with the NIH for non-clinical research using small cell lung cancer patient samples. We are aiming for phase 1 clinical trials of ABN 501 in the first half of 2026. To achieve this, we are conducting studies on the manufacturing process and non-clinical toxicity. Meantime, we are considering a standard of care add-on strategy after confirming ABN 501's potential for combination chemotherapy in clinical studies. Additionally, we are exploring its development potential in various drug modalities such as T-cell engager. The takeaway of ABN 501, which is a Claudin 3 targeted treatment exclusively owned by Abion, a first-in-class drug with the potential to offer new treatment options for various carcinomas, including small cell lung cancer. We are rapidly advancing process development, diagnostic method development, and combination therapy research, aiming for IND submission in the US and Korea by the second half of 2025.
We know that c-Met dysregulation is a real clinical matter in many different diseases. So getting together in non-small cell lung cancer, probably we would estimate the number of patients having MET dysregulation around 250,000 patients, and then the market size may be reaching $5 billion. This is the structure of Babamekib, and Babamekib is highly selective c-Met inhibitor binding to ATP binding portion, and then also enabling phosphorylation further. And protein inhibitor complex stabilize the kinase so that it will ultimately inhibit proliferation of cancer cells. Babamekib has highly selective inhibition against MET, but if you see tepotinib and capmatinib, they inhibit not only c-Met, but also other kinases as well. This actually already predicts that other two TKIs would have off-target effect. Summarizes that Babamekib's selectivity makes the prediction that adverse event profile would be lower than other c-Met inhibitors. aim. First of all, exon 14 skipping non-small cell lung cancer, and then the next will be other c-MET dysregulation in relation to EGFR mutation and resistance to it, and also not limited to non-small cell lung cancer. We have Abamekib's phase 2 trial, Cohort 1 data. Abamekib went through the dose escalation phase 1 study from 50 milligram per day all the way to 1200 milligram a day. And phase 2 is now ongoing in MEDEX 14 skipping population having non-small cell lung cancer diagnosed. Here we can see the response rate. Treatment naive patients, we have seen 6 out of 8 patients so that 75 of response rate. Now, we have actually 35 patients in the Cohort 1 of phase 2. Among those, safety analysis was done in 30 patients. Compared to capmatinib and tepotinib, the actual adverse event was less. But more importantly, if we see the TAE or TRAE above grade 3, a lot less. So 20% versus around 67, 54%, and also 10% of TRAE grade 3 or higher, and 38%, 28%. This actually tells that the profile of Abamekib is a lot safer, and also would be very importantly, the benefit risk ratio will be a lot better. You see the swimmers plot, we can see the median progression-free survival is 11.6 months. This is the case we observed that very rapid response. The assessment was done on the day 16 from the first treatment. As you can see, 32.7% of tumor size reduction was observed. Two key opinion leaders in lung cancer explained that it is very impressive to see the safety profile and then also convincing efficacy as well. Also, the other two TKIs safety profile would support Abamekib data to use in more safely utilized in the patients. Together, in summary, from the phase 2 trial, we would say the compound is very safe and much safer than other two compounds. And then the overall response rate also more or less similar to those two compounds. Because the monotherapy has shown very good efficacy data and then also safety profile, we are moving to the next stage of development. The next cohort in Cohort 2, we are going to develop combination regimen with Abamekib and lazertinib in EGFR mutated non-small cell lung cancer. We have had experiment in two different type of PDX model. So the first model represents in clinical setting failure from the first line third generation TKI. So exon 19 deletion plus MET amplification. Combination, it shows complete growth inhibition. The other model over here, so exon 19 deletion plus T79M mutation. This is actually the model for the second line third generation TKI failure, and then MET amplification. And while lazertinib and Abamekib show moderate or minimal tumor growth inhibition, when it was combined together, it also showed near complete growth inhibition. So we are going to open the Cohort 2 in combination with Abamekib and also lazertinib. We will use lazertinib full dose and also Abamekib 800 milligram. We will also experiment in two different doses to find the optimal dose. And we'll compare standard of chemo versus combination. We will make a good conclusion whether we go for NDA or into phase 3. Now, beyond non-small cell lung cancer, A very interesting case in glioblastoma, and this case was from the compassionate use of the drug. 55 years old female patient had diagnosed as a GBM through surgery. Almost only 2 months later, the patient had recurrent disease. So that second operation went ahead, and a patient had another recurrent disease like here. See the biology of the disease. This GBM had METX963 splice mutation, so METX14 skipping, MET amplification 10 copies, and also the fusion with MET mutate. In 16th stage of treatment, you can see tremendous decrease of the tumor size, around 90% of treatment from the baseline. This is the data from the preclinical in other cancers. So it works very good, not limited to in lung cancer, but also gastric cancer and liver cancer as well. This is another data for combination therapy. It works even better. And very interestingly, when we saw the KRAS mutated and MET dysregulated disease, combination with the sotorasib has shown a lot better result compared to monotherapy each. Conclude that the drug is underway in the phase 2 in METX14 skipping and moving to combination therapy in EGFR mutated lung cancer, and then we would have another possibility in other cancers as well. The real strength of the compound is actually a safety profile with a more or less similar efficacy result. We expand the combination therapy, even only limited to lung cancer, the value of the compound would go to $5 billion. So we continuously will evolve the development of the drug in various MET dysregulated disease. Thank you for coming, and thank you for your participation over here and interest. We really look forward to your continuous support for our development. Thank you so much.